Hello everyone, this is Dr. Pooja, your biochemistry mentor. Now today we will be starting up with a very important topic in biochemistry, enzymes. Now first of all, before starting up with any topic, you should know what is the significance of enzymes in the field of biochemistry plus in the field of medicine. Now basically, what is this biochemistry term? We all know that biochemistry is the chemistry of the biomolecules. Whatever we eat, in what way we eat, what happens to the food when it goes inside the body. So whatever reaction is going on inside the body is catalyzed by enzymes. So anything wrong goes with the body, the enzymes are the ones which are affected the most. So it's like, whenever we start learning a language, for example, if we have to learn English, we need to know the sound, knowledge of the alphabets. For example, we pick any language. So same is the case if we have to understand the core of biochemistry, we need to know about enzymes. So we will be starting up with the first very important topic, enzymes. Now the question is, what are enzymes? Just now I told you they are just the catalysts which are increasing the rate of biochemical reaction. Now you see, it's quite evident from the word itself. Biochemical means the chemical reaction which is taking place inside the body, right? So since they are taking place inside the body, so they become biocatalysts. So how can we define enzymes? Enzymes are simply the biocatalyst which increases the rate of a biochemical reaction. Now what is that reaction? Basically, they will always combine with the substrate. And whenever you have studied in chemistry also, whenever the reactants combine, they are going to collide with one another. That is what the number of effective collisions. More the number of effective collisions, more will be the reaction. And obviously the catalyst is going to increase the rate of that reaction. Same happens with the enzymes. This enzyme will combine with substrate, forming a complex which is known as enzyme substrate complex, where what is going to happen exactly? Old bonds will be obviously broken and new bonds will be produced, and it depends on the number of the effective collisions. So what happens is this enzyme substrate complex will dissociate to form the enzyme plus the product. So basically our main thing, the main criteria is how much amount of product is produced. Similarly, how much amount of product is produced, that depends upon this enzyme substrate complex. So this is the simplest definition of an enzyme, that enzymes are the biocatalyst which increases the rate of a biochemical reaction. I'll just give you one example by drawing a graph. Like if I say this is the normal reaction. Like this. These are the reactants and these are the products. That means reactants have to literally climb this mountain where they will reach the top and then they will descend down. So it depends on the effective collisions that these reactants have to reach here, and this is known as the transition state. Transition state means the state when that enzyme substrate complex will be produced. Now what is happening is we have to activate the molecules, we have to activate the reactants. So what is that energy known as? The energy required to activate the molecules is known as activation energy. So I can simply say that this is energy of activation. Okay. Now this reaction is taking place without enzyme. We can't see any enzyme in it. Now, if we put an enzyme, what will happen? Simple concept. In the presence of an enzyme, the reaction will take place like this. So we can say that activation energy is the one which is decreased. And this red color is with enzyme. So now you can seriously see, what is the difference basically between these two reactions. Without an enzyme, the activation energy is more. In the presence of an enzyme, the activation energy is decreased, and that is what is the function of an enzyme. that enzyme is increasing the rate of a biochemical reaction. The question is how it is increasing? By lowering down the activation energy. Very, very important. And this is the simple concept. Okay. And just now I told you they are the biocatalysts because they are actually reacting with the reactions which are taking place in the bodies. For example, if I say we eat glucose. So how this glucose is converted in the body to release carbon dioxide and water. And you can very well imagine if we are having a diet, say carbohydrate diet all the time, so we cannot eat. So for that particular time, glucose needs to be oxidized immediately. Glucose needs to be broken down immediately. That is why time factor is the most important factor here. An enzyme is simply decreasing the activation energy. So obviously it is going to decrease the time and thereby it will increase the rate of that biochemical reaction. Okay. Now coming over to this, what are the properties of enzymes? This is also very important. How an enzyme acts. So the most important property of enzyme is all enzymes are proteins. Very, very important. All enzymes. This is the most important property. All enzymes are proteins. But there are certain exceptions to this also. Okay, but still they are acting as, again, increasing the rate of a reaction. And the example of this is except ribozyme. Because if you see ribozyme means RNA is having a catalytic activity here. So all enzymes will be proteins except ribozyme. And if we talk about the example of ribozymes, we can simply say that first example of ribozyme is ribonuclease P. Examples are ribonuclease P, very important, snRNAs, and the most important, which you people have studied in detail also, peptidyl transferase. Very important. Okay, so these are the three very important examples of ribozyme. And except these, wherever RNA is having a catalytic activity, rest of the enzymes are all proteins. So this is one very important property of enzymes. And if we talk about the second property, enzymes, since they are proteins, they are heat labile. Now what do you mean by heat labile? Means a slight decrease or increase in temperature is going to denature the enzymes, because we know enzyme is a protein. Protein is what? Proteins as such, they do not exist anywhere. Proteins are simply the polymers of the amino acids which are making them. So whenever we are raising the temperature, whenever we are decreasing the temperature, we are adding an acid, we are adding an alkali, the bonds will be broken. And if the bonds are broken, amino acid, the bonds will break, proteins, they will be denatured, and overall the enzymes will be denatured. So these two are the very, very important properties of enzymes. First is all enzymes are proteins, and second is all enzymes are heat labile. Right. So these two are the important ones. Then if we talk about enzymes, जैसे in our day-to-day life, we are nothing without our friends. Same is the case with enzymes. They also work in an efficient way if they combine with their friends. And what are these friends? These friends can be a metal ion. These friends can help in the form of the vitamins. And that is what we all see also. Whenever you also go and visit a doctor, sometimes he always, recommend vitamin supplements. So have you ever given a thought why he recommend to go for vitamin supplements? Because vitamins are the ones which are helping the enzymes in catalyzing a biochemical reaction. Clear? So this is very important. Now the question is, what is the nature of these friends and how they are going to help the enzymes in their action? So we can simply say that if we talk about the enzymes, if they have inorganic molecule, say for example, if some inorganic molecule is associated with them, we will always say they are the cofactors. Right. And if any organic molecule is associated with the enzyme, it becomes the coenzymes. And you can see from the word itself. The word itself is self-explanatory. Cofactors, working, the factors which are working in cooperation with the enzyme. Enzymes, they are working with cooperation, with that organic molecule. And this is very, very important. You need to know the difference. What are the different types of cofactors? What are the different type of coenzymes? Okay, so I'll just give you one example. First of all, you have to need to understand that what is this enzyme. Suppose if I say this is an enzyme. Okay, so what is this? This becomes the active site of the enzyme because substrate has to bind somewhere, no? So this is the active site where obviously the substrate will bind. So what is this? This becomes your substrate. And this just is like a Pac-Man model which was there earlier in those Nokia cell phones. Pac-Man means it will open the mouth, it will try to engulf the whatever is there, So same thing happens here. This active site, it will be open and the substrate will try to enter inside and it will just close its mouth. And what will be the structure? It is going to form that enzyme substrate complex. So I can say this is the enzyme and the substrate is like this. So this becomes the enzyme substrate complex, which is written in square brackets, means it is very unstable. It cannot stay for a longer time and it will immediately split up into the products. So this is enzyme substrate complex. Now the question is, what is the function of this inorganic molecule? What is the function of this organic molecule? They will just help in speeding up the reaction. Okay. So first of all, I'll just give you the example of cofactors. This is very, very important from examination point of view also, and clinically also this is very, very important. Now, if we talk about the cofactors, I can simply say that if we talk about, let's start with zinc. We all know that zinc is a very, very important mineral which is required again for so many biochemical reactions. Now again it will act as a cofactor. Now what are the reactions in which zinc acts as a cofactor? The most important one is alcohol dehydrogenase. Alcohol dehydrogenase, carboxypeptidase A and B, peptidase A and B, and then cytoplasmic or cytosolic superoxide dismutase. It is a very, very important enzyme which plays a very important role in, you can say, antioxidant mechanism. So these are some of the examples which I have quoted, and zinc, always remember guys, this is the most important cofactor which is present in the body. Then after zinc, we can quote the example of copper. Now, if we talk about copper, we all know that copper is a cofactor for, again, cytochrome oxidase. And the most important enzyme which requires copper as a cofactor is tyrosinase. Very, very important. And if we talk about tyrosinase, if we talk about the clinical application of tyrosinase, absence of tyrosinase leads to albinism, which we are going to discuss in the metabolism of the amino acids. So I'll just write deficiency leads to albinism. So you can just imagine the importance of copper here. Okay. Then after copper, then we can quote the example of magnesium, and we can write manganese also. Now magnesium and manganese, always remember, this is required for the activity of kinase, phosphatase. And if we talk about manganese, manganese is obviously kinase and phosphatase, but the most important is mitochondrial superoxide dismutase. Now here you have to remember one exception. I just want to point it out. Always keep one thing in mind, that magnesium is required for all the kinases reactions except the most important reaction which is taking place in our body, pyruvate kinase. I'll just write it here also, that magnesium is cofactor for all kinases except pyruvate kinase. And this pyruvate kinase is catalyzing the conversion of PEP to pyruvic acid. And here we all know that ATP is synthesized and the cofactor here is potassium. So this is an exception, right? So otherwise, if we say that magnesium is a cofactor for all kinases except pyruvate kinase, where potassium is the cofactor. Okay. So these are the different examples of cofactors. Then after this, then we come over to the coenzymes. Now coenzymes are what? They are simply, you can say, the organic molecules. Whenever we talk about organic molecules, they are the vitamins. So if we talk about the examples of coenzymes, the most important example, very important example I would say is, I'll just make a table here, the name of the vitamin and the function catalyzed by the vitamin. Okay, so if we talk about the vitamin here, the first one is TPP. We all know that TPP, thiamine pyrophosphate, is the active form and it is known as vitamin B1. And if we talk about the function, vitamin B1 catalyzes the oxidative decarboxylation, which we will be discussing in due course of time. Decarboxylation. Simple. If you look at the definition only, it is clear from the word itself. Decarboxylation means removal of carbon dioxide when it takes place in the presence of oxygen. So it becomes oxidative decarboxylation. Okay. Then second is FAD, the active form, FAD or FMN, and we can simply say it is B2. And if we talk about the function catalyzed by B2, we can simply say that it also catalyzes the oxidation and reduction reactions. So I can simply say that redox reactions in general. Right, just for example, if succinate dehydrogenase is there, okay, their FAD is converted into FADH2. Then B3, the active form is NAD or NMN. It is known as B3 or niacin, a very, very important coenzyme, a very important vitamin, and it is in fact vitamin B3 is the most important reductant which is present in the body. And we know that B3 is derived from tryptophan, which is itself an essential amino acid. And this is the basic importance of this. So if we talk about the functions, all the dehydrogenation reaction, you name any reaction where NAD is not used, NAD2, NADH plus H positive. So I can simply say that generally the dehydrogenases or redox reactions. Hydrogenase reaction. Then B4 does not exist. B5, if we talk about, B5 is pantothenic acid and the active form is SH-CoA. And if we talk about the functions, we can simply say that acetyl coenzyme A or acyl coenzyme A or something related to fatty acid metabolism. Then comes B6. B6 whose active form is PLP, which we call as pyridoxal phosphate or pyridoxamine, or you can say simple pyridoxine. And if we talk about innumerous functions of pyridoxal phosphate is there, the most important one is transamination. Very, very important. Then transsulfuration. There is transfer of sulfur group. So transsulfuration is there. Then the most important function, decarboxylation. There is a difference between decarboxylation and oxidative decarboxylation. Oxidative, as it is clear, some extra O is there. What is that? In the presence of oxygen. This is simple removal of carbon dioxide irrespective of the presence of oxygen. So this is the most important difference, and most of the students get confused also in this reaction. So just keep one thing in mind, whenever we are writing simple decarboxylation, जैसे formation of histamine in the body by mast cells of liver, by the decarboxylation of histidine. So this is simply PLP is required. Okay, but when we talk about oxidative decarboxylation, then obviously we have to write thymine pyrophosphate B1. Okay, so these are the three important functions. Along with this, it is required as a component of glycogen phosphorylase. So these are the four very important functions of B6. Then coming over to next is B7. Now if we talk about B7, B7 is biotin. And always remember, biotin always catalyzes carboxylation. Means it attaches carbon dioxide. So you can say that decarboxylation, whenever decarboxylation is there, it is a removal of carbon dioxide. Whenever carboxylation is there, it is addition of carbon dioxide. So it is like increasing one carbon atom. So whenever we are increasing one carbon atom, biotin is used. Whenever we are decreasing one carbon atom, PLP is used. But when one carbon atom is being taken in the presence of oxygen, it becomes oxidative decarboxylation, for which TPP is required. Okay. Then after B7, then we come over to B9. We all know that B9 and B12, these are the two very, very important hematopoietic vitamins, right? So they are actually related with the formation of the blood cells. And B9, B12, it is actually folate. And if we talk about B12, it is cobalamin. And this cobalamin actually exists in again because it's like it is having that Corian ring, and in between a central metal is situated. So if that central metal is methyl, it becomes methyl B12. And if it is adenosyl, it becomes adenosyl B12. Okay. And if we talk about folate, the most important example of this folic acid is it is responsible for 1-carbon metabolism. 1-carbon metabolism is actually the backbone of the most important reactions in the body. As it is clear from the word itself, 1-carbon. Mane ye kya karega? It will always give 1 carbon. Whether it is in the form of methyl group, whether it is in the form of methinyl group, whether it is in the form of formyl group. So whenever 1 carbon is given out, it becomes 1-carbon metabolism. And folic acid, tetrahydrofolate, dihydrofolate, they are the active forms of this. They will always give 1 carbon. And reactions we will discuss again in different topics which we are going to cover in the later videos. And if we talk about, for example, a most common reaction, I'll just tell you of folic acid is, say, whenever uracil is converted into thiamine. Okay, uracil is the base present in RNA and thymine is the base present in DNA. And the most example, whenever a virus invades the body. So what happens is, virus, we all know, it lacks a host for replication. So the moment it starts invading the human body, it tries to capture the host machinery. So in that bargain, it increases the production of thymine. So for that, again folic acid is used. So what we do is, the most important cancer therapy is we have to actually search for folate reductase inhibitors. And most of the anti-cancerous drugs also act as folate reductase inhibitors. Okay, so I'm just trying to give you the concept behind this 1-carbon metabolism. And if we talk about cobalamin, mein methyl cobalamin and adenosyl cobalamin are the two examples. And if we talk about the reactions, the most important reaction, it is required for methionine synthase. And second important example is, along with methionine synthase, methyl malonyl coenzyme A mutase. Both the reactions are very, very important. And वैसे भी just keep one thing in mind, B9 and B12 are just, like best friends also. It's like B9 and B12 are interdependent on one another for only one reaction in the body. But yes, there are certain point of differences also between B9 and B12, but there is one reaction which just cannot survive without B9 and B12. So these are the examples of the coenzymes. So you can just imagine that the vitamins which are functioning as coenzymes and the metals which are functioning as cofactors, they are helping the enzyme in their function. So this becomes the property of the enzymes also. And here one more thing, cofactors mein, this is like the metal which is requiring. Again, one more example here is that calcium is also required for lipase. Lipase, which helps in the digestion of the fats. Right? So these are three very, very important properties of the enzymes. Okay. Now, if I talk about this, I can simply say that this enzyme is basically having a protein part. Just now we saw protein part. And along with that protein part, it has a non-protein part. This non-protein part is actually the prosthetic group. Okay, which can be either a cofactor or a coenzyme. Right. Now the question is just the protein part of an enzyme, this much part. Protein part is known as a apoenzyme. So if somebody asks you what is an apoenzyme, apoenzyme is simply the protein part of an enzyme. But when this protein part combines with a cofactor or a coenzyme, it becomes holoenzyme. Very, very important again. So if I ask you to differentiate between apoenzyme is just the protein part. But yes, holoenzyme is the entire thing, which will comprise of the protein part along with a cofactor or a coenzyme. Right. Then after this, then we are moving on to the last property of the enzymes, that enzymes are highly, highly specific in action. Specificity of enzymes. And if we talk about the specificity of enzymes, no matter what happens, enzymes are region specific, enzymes are absolute specific, enzymes are stereo specific. In a nutshell, enzymes are highly, highly specific. So I can simply write that enzymes are highly specific. Right? Whether it is stereo specificity, whether it is absolute, or if we talk about region specificity or regiospecificity, means example is, say, if L-amino acid oxidase is there, right, it will act only on L amino acid, not D amino acid. Right. So these are the 4 very, very important properties of enzymes. Then after this, we need to know about the definition of enzymes. That, just suppose if I am taking a class of say 100 students, and if all the 100 students pass in the class of biochemistry with good marks, so it's like the turnover number of the class would be very good. And suppose out of those 100 only 10 pass, that means the turnover number, the overall result will be bad. Same is the case with enzymes, because enzymes are the ones which are obviously catalyzing the biochemical reaction. So the body just cannot afford to miss the reaction for even a fraction of a second. Okay, so this is the most important property, or you can say this is the most important characteristic of enzymes, that enzymes are having turnover number, which is actually known as the catalytic property of an enzyme. And how do we define? We simply say that Kcat will be equivalent to the maximum velocity over the total enzyme concentration. ET is the total enzyme concentration. Right, and if we have to define it, we can simply say that catalytic efficiency will be defined as Kcat over Km. Again, this is very, very important. The catalytic efficiency of any enzyme will be equivalent to Kcat, which is again the catalytic constant, or which explains the turnover number of the enzyme, over Km. Km is simply Michaelis Menten constant, which again we will discuss when we are going to discuss again the kinetics of enzyme. Right. So this is again very important. Then after this, then we come over to the next subtopic, that how the enzyme binds with the substrate. The question is how the enzyme bind with the substrate. So there were 2 theories which were put forward, that how the enzyme is reacting with the substrate. So first theory was lock and key theory, and second was induced fit. Lock and key and induced fit. First of all, you need to understand what is the basic difference between these two. So this lock and key theory was given by Emil Fischer. And induced fit was given by Koshland. Okay. Now as the word suggests, lock and key, जैसे we normally say ना, since every lock is opened by its own key, so every enzyme, since it has an active site, so every enzyme is going to release, it is going to open the active site only for the specific substrate. Okay, so obviously just now I told you that the enzymes are highly specific about the substrate. If there is any slight change in the substrate also, they will say no, that they are not going to bind with it. But to some extent it is possible, but whenever we talk about the biochemical reactions, whenever we talk about the actual property of enzymes, which are proteins, so enzyme has to be specific, but yes, to accept the specific substrate, it tries to accommodate the substrates. Why? Because it is again nothing but a protein. So a slight change is affordable. Okay, but Emil Fischer was unable to diagnose it. And he simply explained that the enzyme is very rigid. Suppose if the enzyme is not matching up with the substrate, with the active site, it will say no. Whereas Koshland was the one who gave this concept that since enzyme is a protein, there has to be a change in the structure of that enzyme so that it should accept the specific substrate. Okay, so I can simply say that lock and key basically approach the rigidity of the enzyme. But if we talk about Koshland's induced fit, it explains the flexibility of the enzyme. So you can very well imagine which theory will be widely acceptable. The theory which was widely acceptable was Koshland, who explained the flexibility of the enzyme, which simply means that even if the enzyme has to change itself, it is going to accept the specific substrate by, it can even try to accommodate, it can even change the conformation. Whereas lock and key, simply rigidity, if it is not suitable, it will say no. Okay, so I'll just write it down, that rigidity, so it actually failed. So this is the basic difference between lock and key and induced fit. Lock and key explained the rigidity of the enzyme, so basically it failed also. Whereas flexibility of the enzyme, it was approved and it is widely accepted also. So this is how we can say that these are the two important, very important, I would say, how the enzyme is binding with the substrate. Okay. Then after this, then we come over to the next important point. Just now I told you what is this enzyme activity, how active an enzyme is. That means, how can we explain the activity? How an enzyme is going to react with the substrate in a particular period of time so that it can form maximum amount of the product. Now the question is, since enzyme is catalyzing the biochemical reactions in the body, what are the factors on which the activity of the enzyme will depend? So next is factors affecting the enzyme activity. Very important. Now, the important factors which can affect the activity of the enzyme include the temperature, very important factor, pH, hydrogen ion, negative logarithm of hydrogen ion concentration, obviously the substrate concentration, which plays a very, very important role and on which actually the entire kinetics of enzyme depends. Substrate concentration. So these are the factors which will affect the activity of the enzyme. Now, first of all, we will take up the first factor, which is temperature. Now, we all know that enzymes are actually catalyzing the biochemical reactions in the body. So, you can say the normal temperature of the body would be 35 to 40 degrees centigrade. So, how the temperature will affect? A slight increase in the temperature, a slight decrease in the temperature is going to denature the enzyme. Okay, so obviously it is going to influence the activity of the enzyme too. So, I can simply say that temperature may the enzymes show maximum activity between 35 degrees to 40 degrees centigrade, or this is the optimum temperature or the temperature optima. And if we have to draw a graph, here we can write the temperature here, and here we can say the enzyme activity or the velocity of the reaction, whatever. So if we talk about the influence of temperature, we can simply say that it is going to attain a bell-shaped graph like this. And if you see, this is the maximum temperature optima, or I can say that 35, 36, 37, 38, 39, 40. And this is the optimum temperature. And the graph so obtained is a bell-shaped graph. And you can say that a slight increase or decrease, increase or decrease is going to denature the enzyme. Right. Then second factor, if we talk about, it is the pH, or we know that pH is negative logarithm of hydrogen ion concentration. So if we talk about the pH, and again, जैसे if we are talking about the temperature optima, there has to be a pH optima also. So all enzymes will show pH optima between 5 to 8. In some books it is written as 9. So it is little bit towards acidic, little bit towards alkaline. Okay. But there are certain exceptions here. And the most important exception here is if we talk about pepsin. Pepsin works at pH 2, which is very highly acidic. And if we talk about ALP, which is alkaline phosphatase, which is a marker enzyme of bone diseases, here it works at pH 11, a very high alkaline pH. In fact, most of the digestive enzymes, trypsinogen is there, chymotrypsinogen is there, they always work at a very high alkaline pH. Okay, but overall if we talk about, all enzymes will show a pH optima between 5 to 9. Same graph will be obtained as in pH. This is again the enzyme activity, and here we are going to write the pH. So we can say 5, 6, 7, 8. So this becomes the pH optima. Okay, and again if we talk about the graph, it is bell-shaped graph. So these are the two very important factors which are affecting the activity of the enzyme. Then we come over to the third factor, which is again very important, which is substrate concentration. Now if we talk about this substrate concentration, we know that enzyme is catalyzing which reaction? The reaction catalyzed by enzyme is enzyme combining with the substrate, forming enzyme substrate complex which dissociates to form the enzyme and the product. Okay, so if we talk about, just now I told you that we have to basically concentrate the amount of product produced. Okay, and how we come to know that how much amount of product is produced? We have to see how much enzyme substrate complex is produced. Okay. So obviously, and I told you that enzyme is going to accept the substrate at the active site. More the active site, more will be the substrate reaction, more will be the enzyme substrate complex. But a stage will come when all the active sites of the enzyme will be occupied by the substrate. What happens then? No active site is left for the substrate to bind. So it is going to attain a saturation state kinetics. This is very important. So I can simply say that if I have to draw a graph also, you can simply say, see that Here we are going to take the substrate concentration, and here again we are going to take the enzyme activity or the velocity of the reaction. Initially, more the active site, more will be the enzyme substrate complex. But when a stage comes when all the active sites of the enzyme are occupied by the substrate, it attains saturation kinetics like this, right? So I can simply say that this becomes zero order kinetics. And this becomes first order kinetics. Or we can say that Vmax by this. Okay. So if we have to explain it, and this graph is known as hyperbola. Hyperbolic graph will be produced. Unlike in case of pH and temperature. In pH and temperature, the graph obtained was simply a bell-shaped graph. But when concentration of substrate is there, it forms a hyperbola. Okay. So I can simply say that more the active site, more is the reaction, right? But when all the active sites of the enzyme are occupied by the substrate, it attains saturation state. So it attains a saturation state, right? It attains steady state kinetics, and it attains zero order kinetics. So you can say that this is the most important point which we have to remember, which we have to understand also, that initially when more is the active site, more will be the reaction, more will be the enzyme substrate complex formation. But when all the active sites of the enzyme will be occupied by the substrate, it is going to attain saturation, steady state or zero order kinetics. And the graph obtained is a hyperbola. And this concept, and if we talk about zero order kinetics, the most important thing is that both are independent of one another. Means substrate enzyme becomes independent of one another. Enzyme are independent of one another. So these are the three main important factors on which the enzyme activity depends. One more is there, you can say enzyme concentration, more the enzyme, more will be the reaction. So it has to be straight line. But these are the three which are generally considered and very, very important. Okay, then after this, then we come over to the next topic in this again, because on the basis of this enzyme kinetics was given. Now, if we talk about enzyme kinetics, we know that what is kinetics? Kinetics is simply that how an enzyme is combining with the substrate, what happens when an enzyme combines with the substrate, and basically in the end what is produced. So kinetics is there is an initial velocity, there is a maximum velocity. Okay, and if it again look at the reaction, I can simply say that enzyme will combine with the substrate. It will form enzyme substrate complex which will dissociate to form the enzyme and the product. So here if I say this can be K1, this will be K minus 1, this can be K2. I can say that K1 is a rate of formation of ES. If I say K minus 1 is a rate of dissociated ES, or you can say rate of ES which is not converted into product. Converted to product. And if we talk about K2 is a rate of formation of product. Right. And this kinetics was very well explained by two scientists, Michaelis and Menten. They were the two scientists who gave the concept of enzyme kinetics. Michaelis and Menten. And what equation they gave? They simply said that V0 is equal to Vmax S over Km plus S. This was the equation given by Michaelis and Menten. And as per this equation, we can simply say that V0 is the initial velocity. Vmax is the maximum velocity. And if we talk about S is the substrate concentration. And Km is Michaelis-Menten constant. Menten constant. So this was the equation given by Michaelis and Menten, and it explained simply the kinetics of the enzyme. Now the question is, and just now we saw that it gives a hyperbolic graph. Now the question is, which is very, very important, what happens when initial velocity is half the maximum velocity? What happens when initial velocity is half the maximum velocity? V0 is equal to Vmax by 2. And if we put this in this equation, V0 is equal to Vmax S over Km plus S. And if we calculate it, it comes out to be Km is equivalent to the substrate concentration. So I can simply say that whenever initial velocity, very, very important in fact, when initial velocity is half the maximum velocity, Km is equivalent to the substrate concentration. And this is what is the importance of Km also. When initial velocity is half the maximum velocity, Km will be equivalent to substrate concentration. Now the question is, this is how we derive. What is the significance of Km? Why we have to do this much of procedure? So the significance of Km is it is inversely proportional to the affinity of the enzyme with the substrate. So I can say that Km is inversely proportional to the affinity of the enzyme with the substrate. Inversely, so I can say that if any enzyme has high Km, any enzyme, so obviously the affinity would be less of the enzyme with the substrate would be low. And if we talk about any enzyme which has low Km, the affinity of the enzyme with the substrate would be more. And if we talk about the ideal substrates, ideal substrates also always have low Km or high affinity. Very important. And this is what is the significance of Michaelis-Menten equation, where they simply said that Km is inversely proportional to the affinity of the enzyme with the substrate. And if we talk about the examples here, we can simply quote the example of hexokinase and glucokinase. And if we talk about hexokinase, hexokinase has low Km and glucokinase has high Km. So obviously if hexokinase has low Km, affinity is high. And if it has high Km, low affinity. And that is why we always say that glucokinase is present in the liver, hexokinase is present in all the tissues. Whenever a shot of insulin is given to a patient suffering from hyperglycemia or DM, automatically insulin is going to act literally on glucokinase, thereby increasing the uptake of glucose by the cells. So that is what is the role of this Michaelis-Menten equation. Okay. Now coming over to this, that this was the concept given कि when initial velocity is half the maximum velocity, Michaelis-Menten constant is equivalent to substrate concentration. But there was one more scientist who gave the concept of, I would say कि he wanted to actually convert this equation into an equation of straight line, just to predict what would be the slope and what would be the intercept. And that scientist was known as Lineweaver, and the equation which he gave, the graph which he gave, it was known as Lineweaver-Burk's plot. Okay, so after enzyme kinetics, then we come over to the Lineweaver-Burk's plot, which is nothing but it is the reciprocal of Michaelis-Menten equation. So I can simply say that Lineweaver-Burk's plot is simply the reciprocal of Michaelis-Menten equation. Michaelis-Menten equation. So I can say that this is Michaelis-Menten equation. V naught is equal to Vmax S over Km plus S. Right? So basically we have to convert it into 1 upon V naught is equal to Km over Vmax S plus 1 upon S. Km Vmax S, S over Vmax S आ जाएगा. Okay? Or I can simply say that Vmax like this. Okay? And this will be converted into Y is equal to Mx plus C. You can do it in your home also. Simply what I have done is I have just reciprocated this equation. So Km Vmax S, S over S bata Vmax. Okay. And if we plot a graph, it comes out, this becomes the slope, SS will cancel and this will become the intercept. So I can say that slope will be Km over Vmax and intercept would be 1 upon Vmax. And if we plot a graph, the graph will be like this. Since it is a straight line graph, this is 1 upon V naught, this is 1 upon Vmax, and it will be like this. 1 upon substrate concentration, second 1 upon. This will be your Vmax or the intercept, and this will be the slope. So I can say that this will be the slope and this will be the intercept. So this is Lineweaver-Burk plot, which is a reciprocal of Michaelis-Menten equation.